Nynke Dekker. I'm professor of biological physics at present, both at the University of Oxford and TU Delft. And our research is really on the use of single molecule techniques to study biological processes. These days, the focus is primarily on DNA and RNA replication. These days, we can look at more complex biological systems. DNA replication is carried out by the replication, which is a multi-component machine. So we'd really like to understand how it works as a machine, figure out the parts are kind of transient or can be transient. So we'd like to see how transient, right? Which ones are where, when. And then the replication operates in this context, which is biologically complex, right? There can be other proteins bound to the DNA. There can be damage to the DNA, right? How does the machine then also recover from that? So really trying to understand both the dynamics of the replisome in a simple context on just bare DNA, but then also in the context of sort of either problems on the DNA, if you will. And that can also include chromatin. Over the years, I've become more and more of a fan of biochemistry because it's super inventive and can look at, there's so many methods that are associated with it. And definitely they can learn many things, right, about things like DNA replication. But the same thing holds true for these days for cryo-EM. That's also a methodology that is very active in the field. But of course, the ensemble biochemical methods, they do averaging, right, over many, many molecules. And so does the cryo-EM, even though ultimately that's what allows it to get the very high resolution. But what you're going to sample is still, what's going to really be visible in your data is still an average state. And of course, in single molecule, you don't have that, right? You look at the individual molecule. Right, that allows you to then also get a better view of its dynamics, knowing what an individual replisome is capable of and also not capable of, or how it needs to react in case there's a problem. That's relevant. You can't only just measure the average. In general, I would say the ease of use is high. You can get started very quickly, and if you have a good person training you, I think you can advance rapidly. I think in general, it provides a way to get good plots, and anything that we can do to get people more quickly towards good science, I think is a good thing, because it'll mean that the conclusions that people draw from their data will be more solid. So in the end, it will help knowledge. Generation of knowledge in general. For us, this work was really like a stepping stone towards really establishing, let's say, CMG on a chromatinized origin, and then looking at downstream chromatin replication events. And there, of course, at some point, your bulk experiments will no longer be able to report on that, right? Yeah, I think assessing mobility at some point using bulk assays becomes a bit limiting. And so hopefully that's an avenue that we as a lab will be pursuing using single molecule techniques in the coming years. And